sure about one thing that I will not be doing my PhD in India. That is something I'm certain.
Okay, that's a good start.
Yeah. Okay. So let me begin. Uh, Welcome to my podcast, Nikita. In the second episode of the Compare podcast, with us we have Ms. Nikita pursuing her PhD at the University of North Carolina at Charlotte, and she's the president of the PhD Career Ladder Program, an initiative that helps PhD scholars to explore and prepare for their career. Welcome, Ms. Nikita.
Well, thank you so much for inviting me. I'm really excited to talk about my journey and like hopefully, people will learn something from it, from my experiences per se.
Sure. So, you started with a BSc in biology at Delhi, and right now you're investigating how chaperones are overexpressed in cancer cells. So, how did
Mhm.
this actually happen? This big changeover?
So, just like all the undergrads, like I did not have any idea what to do with my life after like bachelor's. So, like after high school, basically. So I started like bachelor's in zoology in Hansraj College at University of Delhi. And so zoology was like full on dissections. I used to dissect rats, mice, prawns, fishes, Drosophila. So it gave me a an exposure to what I could do in terms of like in the field of science. And that's when like I realized I really want to go for higher studies. So I applied for masters in zoology and I was the topper of my class in bachelor's of science. So my teachers of course were really encouraging and they encouraged me to pursue a career in like masters in zoology. So I decided to do that, and once I went to masters I learned a lot more as well in terms of like what's all out there. So we were taught bioinformatics and genomics, so it really gave me a good exposure. Then I learned about like PhD program because of course our department has a PhD program. I saw like PhD scholars there, and I used to talk to them about like how their life is as a PhD. So most of the PhD students were like they were happy in their program researching. Some of them wanted to go for lecturership position, but as far as like I gathered, I wanted to do something in research other than like also like teaching, more than teaching, I should say. So one of my teaching assistants was really encouraging and motivating in terms of, he was like, you should try and apply and go abroad and see what's out there. It's better than here. Of course, like once they are in PhD program, they know like what's out there. And me being a new student, I did not have much of an idea. So he really encouraged me. So initially I was like, no, I want to do PhD in India. And I was like, maybe I should try for JRF. And like I scored like AIR 44 in JRF, uh, CSIR JRF. And then like I also got like ICMR JRF. So I
That's a
started
big
working in
thing.
a
Really
thank
competitive.
you. Yeah. So I started working in a lab as a project assistant, and the lab was basically epigenetics and chromatin biology. And that gave me a lot more exposure to research because now I had my own project and I was working on this chromatin remodeler INOAT and its role in, cancer cells. So, but I wanted to do more and I wanted to do a PhD. But before that, I should also mention that I also got offered a PhD at National University of Singapore uh, while I was doing masters because I was topper of my class. But back then, I was not so sure that I wanted to leave my family and go to, Singapore. But like you see, years later, I'm in US, so that's like weird. But I think exposure to that research as a project assistant in University of Delhi in that lab for a couple of years gave me really a chance to like explore, science really well because now I had my own project. I could see like what's out there because most of the PhD students who graduated from the lab were going abroad for their postdocs. And when I used to talk to them, they were really excited and really encouraged me like it's better here, the research facilities are really good here as well. So you should try your hand at like joining PhD in some of these labs. So then, like, I started applying for PhD positions abroad. I was open to Europe or US because, like, I know, like, they are at par in terms of like facilities. So I did get interviewed at Institute of Pasteur in Paris, but like, I did not like, you know, make the final rounds. I did like Hanover University. Again, like I reached the final rounds, but I did not give up. So then I started applying for US. I gave my GREs, I gave TOEFL, and then I applied to a couple of labs. Uh, so I got offered, I should say, I interviewed, I think, at least at 10 places, and I got offered four positions in like four different universities. And that's when I decided to join Dr. Truman here at UNCC because his work was overlapping with what research experience I had, and like I knew what I wanted to do. And also, like, he is a young PI and he's really encouraging and motivating. So I decided to go with uh, his lab. And also, I'm his first PhD student, and it has been
Oh,
really great so far. So I think like all in all, I should say like that wait was worth a while because now I feel that we are a perfect match. Uh, and I have published like 13 publications so far, and he's really motivating, encouraging in terms of whatever career I want to pursue, and works with me over like what I'm interested in, and like also like if I have some ideas, we work on that together. So it has been really great so far. So I would say from this, the gist for young students who are applying is like never give up, keep on trying, and like you cannot see it happening right now, but that wait would be worth a while, and one day in the future you'll realize that yes, that was worth it.
yeah, that's true. And so you told that you had interviewed for 10 labs and you were selected for four out of them. So, like, did you undergo any sort of lab rotations or you were directly placed in the project in Dr. Truman's lab?
So in our university, we do have a lab rotation program, but like I did not undergo a lab rotation because I knew I wanted to join his lab. So I just like went for it.
Hmm. So,
But like many
the
universities do have like a lab rotation program, which is like beneficial to people because like they can know like what lab they want to join. But I think like this worked out for me and I did not want to rotate in any other labs. So there is that.
and some graduates even consider that a waste of a year.
I guess you could say that if you know where, if you don't, if you know where you want to join, but you still have to do lab rotation. But like if you don't know about the PIs in the department, it's better to do a lab rotation so that like, how you will fit into that lab.
Yeah, so you'll understand how the PI and the relationship between yourself and the PI works. So a lab rotation would be useful in those cases. I agree.
I mean, yeah, that's true. But like, sometimes you go to a lab and you immediately fit in, but sometimes you go to the lab like but you're not able to like communicate. I won't say communication, rather I would say like you don't see yourself fitting, or like your work style does not match your PI's work style. In those cases, like I think lab rotation is important so that like you have an exposure to different labs, and then like you fit in wherever you want to, like at the end of the day, like where you see yourself in 5 years, because again it's a long commitment, and this has to be like a well thought of decision.
Yeah. So apart from your ranks and you being your university topper, what are the kind of skills that you possessed that made you stand out from the rest?
So I had a lot of lab experience as a project assistant, so I had worked on like basic molecular biology techniques. Like I knew how cloning, Western blotting, and let me see because I cannot recall. Yeah, so I had like experience with Western blotting, cloning, real-time PCR. I did chip, seq, ELISA, immunoprecipitation. And in terms of mammalian cell culture, I knew how to do transfections and protein extraction and stuff. So I think like when I talked to Dr. Truman back from now, he was like, yeah, that those were the skills I was looking for. So one of the things that I did not mention is like I contacted him before like applying to his lab.
Hmm.
So that's a good thing. If you are interested in a lab, you should contact the PI and be like, hey, I'm interested in your research. This is my CV. What do you think? So one of the things that like really struck me for from him was like when I of all the places, like of all the PIs I had applied on, he was the one who was the most encouraging. So I just wrote to him, hey, I am this person, I'm interested in your lab, I have this experience, this is my CV. And he gave a really, nice reply. He was like, that's great, and you are the perfect match for me. Like, I am looking for someone like you. Feel free to apply to the PhD program. So then I like, yeah, so for PhD application, you have like couple of things. So you have like your statement of purpose where you
Yeah.
basically like write like how you are a fit or why. It's basically your sales pitch. So you have to tell you like what is good in you that they should buy.
Yeah.
So you have to write like what all like the skills you have, and like on that thing you can mention like the PI you're interested to work with. So that's what I did. So I asked him, I want to apply to your lab, and then in my statement of purpose I wrote his name as a potential PhD supervisor. And then after I submitted that, I was like, hey, I applied to your lab, and then here you go. This is my application. And he was like, okay, I'm going to look out for your application. So that's another way to make your application stand out. Contact the PI beforehand. If they say yes, just apply for the program. And that's how they know that you are the person they should be looking for because you're applying to their lab. So you have to make yourself stand out. I mean, I hate to say that, but like you have to do that.
And that is
Because
a bold decision from your side because in most cases, a graduate student would not prefer to join a new PI, a young PI who has no PhD scholars. But you've taken
okay,
that
so
decision
yeah.
and you've successfully published 13 papers here. That's something great.
So I was also, when I talked to people about, of course I had four offers, so I was confused where to join. But like for some reason I knew I wanted, because sometimes you have that feeling, okay, I think this is right. So I had consulted several people, and they were like, it's a risky decision because he's a new PI, he doesn't have a grant, and you don't know what's gonna happen. So I was like, I'm just gonna take a risk because you have to take risk in life, and sometimes like it's just the gut feeling that tells you like this is right for you. So that's actually what happened here. So yeah, you are right, like people did tell me that he's a new PI and like it's risky, but what, I took the risk, and I think like it was worth a while. So while I'm here, during my duration here, he got two R15s and he's about to get an R1. So basically like he has accomplished like a lot, and he has got like three grants. It was like he had faith in me and I had faith in him, so it like worked both ways.
The mutual trust that every PI and PhD grad is the mutual trust that is needed for every pair. Agreed.
That is true.
Yeah. So, and from what is listed about you, and we found that you are the president of the PhD Career Ladder program. So, what made you join this, and how is this impacting the life of PhD scholars right now?
Okay, so this PhD career ladder, basically, first of all, let me tell you what this is about. So in our department, so grad students basically know like about academia. People are not well, like aware about positions other than academia. So where you can go after your PhD. That's like another big question that people have. I have a PhD degree, what to do? Should I go for a postdoc? Should I do like industry position?
Industry.
But yeah, what we don't know is there are a lot of positions other than like just, postdocs. Because again, like I'll talk about US. In US only, like the percentage of people from postdoc to academia or making it to like assistant professor is 2%. So
Hmm.
that's yeah, very less. So
Very
you
less.
have to think about like career options, and also like if you are not if you have PhD degree, I know like people like to do science, but like if you don't want to like be in academia, face that pressure because it's very competitive, what can you do with your degree that will still help you like use your research skills, but you are like doing something you love. So that was the basis of this PhD career ladder program. So I, along with my lab mate, Laura Knighton, so we started and co-founded this club called PhD Career Ladder Program, where we basically tell students what is out there. So what we do is we have in experts from various industries. So let me tell you what you can do with your PhD degree. You will be mind blown because I was too. So you can go to industry, you can do a research and development position as a scientist. You can be a scientific consultant. You can be a patent law officer or patent officer. You can file patents. You can be project manager in industry. You can lead clinical trials. You can be a clinical trial manager, and so on and so forth. So this list is like endless. So you have a
Endless.
lot of options. Yeah. You can do a lot of research based work, or like if you don't want to do research based work with your PhD degree. Because like Laura did not want to do like, academia. So she was like, I want to do something where I can use my research skills and like still, uh, get a job with my degree. So we started this club. We invite like people from industry. So we invite like scientific consultants and have information interviews with them, just like you're doing with me today. So to gain the insight and see like how they reach their position, like where they are today and what it takes to be there. So that's what we started. And I should say, like, we have been really successful at that because, like, with that, starting that, like, last year, Laura has already gotten a scientific consultant position in a consultancy firm. We had another student who just joined US patent office as a patent officer. So, and this is like still continuing, and like now I'm leading the program. So we invite like people from industry or like different career options each month, like, and they give us insights and how they, reached the position where they are today. And there are people just like us. So they did just their PhD, their masters, their bachelor's, masters, their PhD, and then they didn't want to go to academia either, so they decided to follow this alternative route, and they are successful in their position. So that's what this club is about, and I think like this was really a good move from our students, because like and when this came from a need of like people not knowing like what to do next in their after their PhD. So we started this initiative, and this has been going really well so far.
That's a really great initiative, and we would like to appreciate you for this initiative because that would have changed many people's mind and in turn their career options, making better decisions.
Yeah,
Congrats on
thank
that.
you. And thank you. I think so too, because like uh another thing is like if you don't like, so there are type of people who like people have analytical skills that, but they don't like wet lab work. So how they can use their analytical skills that they learned during their PhD. So again, let me tell you, like PhD is just not about like wet lab. It's about like developing your analytical skills, developing your research abilities. So it's an what I like to say, it's a wholesome experience. So you change as a person in your PhD because like you develop patience, because sometimes your experiments won't work, sometimes they would, sometimes they won't. So you have to have patience, okay? And then things don't work, so you have to figure out why they are not working. So there comes in your troubleshooting. So you develop your analytical abilities, you go read literature, and you find out like, have other people faced the same problem, or is am I the only one, and if so, why is it happening, and what can I do to change it? So that's why people like seek out candidates with PhD, because they have like these abilities to uh, troubleshoot, have analytical abilities. So that's why it's important for like you would think like, well, I can get these jobs as a master's student, but no, like PhD is like entirely different process, and people do seek out for PhD candidates for these reasons because you have all these skills which other people don't. So you stand out in terms of that.
And coming to PhDs, like there are two basic kinds of PhDs. You either get placed in a project, and there is this other one wherein you work for an industry sponsored PhD. So,
Mhm.
would it be better to choose an industry oriented PhD so that you kind of get the extra benefits like what you mentioned previously?
I think like if the goal is to be in industry after your PhD, that would be really great if you can get like a PhD in industry. But also like I am in academia, I'm not sure how that works. From what I have heard is like it's kind of an independent. You will have some help, but like you will have, like senior scientists you are placed with. But I think that could work for people who want to stay in industry, and it would be really, better, and like you will have a smoother transition rather than from going from academia to industry. Because I'll tell you that I hold informational interviews with people from industry like Pfizer and like Novartis. What
Hmm.
they want is they are looking for, sometimes in the job adverts they are looking for people with industry experience. So what they told me is like industry experience is important because like academia is similar to industry, but industry is more rigorous in terms of like results and reproducibility of results, and like it's an entirely different type of process because everything there is transparent as well. I'm not saying it's not transparent in academia, but industry is very rigorous because like they're spending a lot of money on you, even like in terms of like, salaries. And they have, first of all, they have unlimited resources because of course, like it's a privately owned industry and they have a lot of money. So and they are very rigorous, and there is a certain kind of mindset that people have there. So that's why they want you to have an industry experience. So I think all in all, if your overall goal is to go and like have a position in industry after PhD, that PhD, industry sponsored PhD, is a well suited thing. And I think I have seen a lot of adverts from like Pfizer and stuff where they do like industry PhD.
Hmm.
So if someone wants to do that, just look out for these companies like on LinkedIn, like Pfizer, Novartis. They do have these programs for industry PhD. I think AstraZeneca has one. So these are, first of all, also they are very competitive to get in. So again, like if you want to join these programs, contact the people who are already there and see like what they're looking for, whether you're competitive for that PhD position or not, or what do you need on your CV that would make you stand out from the rest of the applicants.
Yeah. So that was some good insight from your side. And we would like to hear about your research experience. So we know that you work on chaperones and their expression in cancer cells. So what exactly is your research?
So again, you talked about like projects. So I've worked on multiple projects throughout my time here. So I started with, so let me just begin by saying that the project you start with isn't the project you're going to finish with. So
Hmm.
do not lose hope. Your projects can change during the course of time depending on whether like they, lead somewhere or not. So I started with like, I think a project that it started working a little bit, but then like we did not know like what direction it was going. So we had to like stop it right there. And then like I started working. So how my PI works is like we have one main project, which will be a big paper, and then we have multiple small projects. So the idea behind that is like as a PhD student, you cannot take a risk. You have to graduate with thesis, right? So you need a project that, you know, will help you in successful completion of your dissertation thesis. So I really like the strategy that my PI takes. He has like one big project, which is like, a lot of work and like a bigger picture. And then you have multiple small projects, which, are small and easy. I won't say easy, but I'll say they are like smaller. And then,
Comparatively easier to
well,
complete.
nothing is easy in science, so, but these are small. So we publish them like, uh, published.
Often.
They are
Yeah.
publishable like, in mid-level journals. That's what I would like to say. So that's a really great strategy, and I think that has worked so far for everybody in the lab. So other than me, he had like another PhD student and then a master's student, and people are joining because now that I'm graduating as well, like hopefully next year. So I worked on a couple of different projects. Now I would have to look at my papers to see like what projects I've worked on. So the very first project I started was like it was a small project. This was tagging like heat shock protein 70 isoform using CRISPR-Cas9. So we basically use like epitope tag to insert this segment into the genome, and then now you have like a tagged HSP70 using CRISPR-Cas9. And I think that was the first paper that I published in the lab. So that's what I started working on like as soon as I joined, and like 1 year later we got that published. So that's in the bank. So we have one publication to say yes, like I have worked on this project. And then also we have a collaborative environment. So all of everybody works with everybody, so we are collaborative. So you are the lead on the project, but you also contribute to the project. So that makes sure that people have like, uh, so that increases the chances of you getting a paper. And I think science is all about collaboration. And if you are collaborative, then it will be like you will be more successful. So then we had like projects with undergrads that I contributed on, and then I think my main project was one of that just got published last month, I should say September. So this was basically a chemogenomic screening that identified one of the helper protein of HSP70, which is HDJ2, as a hub for anti-cancer drug resistance. So what I did there was we generated this CRISPR knockout of this gene. And then we screened this cells which had this CRISPR knockout for anti-cancer drugs that are used in clinic right now. So what we were looking for is the drugs that would, help the cancer cells die. Basically, not help. I should say that they are killing cancer cells better. So what we found in our screen were there are a set of drugs that kill cancer cells. They are like sensitizes like cancer cells, but there are set of drugs that make cancer cells grow better. Now, this was surprising to us because like we did not expect this. That you are adding because normally what happens is like you add an inhibitor and cells die, right? But like why would a cell live when you and it's growing faster or better? So this basically led us to like investigate furthermore like what's happening. So because like in normal humans or like patients or cancer patients, you cannot, administer CRISPR as of right now, but maybe you can in
Ethically.
future. Yeah.
Yeah.
Maybe you can like, modify a gene and like, but like you cannot do that right now. So we wanted to see whether this like replicates with the inhibitor of the helper protein, and we wanted to test whether like this actually is a real thing in prostate cancer cells. So that's what I did. So we had these inhibitors in combination. So let's suppose I'll take an example of VEGFR inhibitor. It's known as cabozantinib, and it was found to be synergistic. That means it was helping cancer cells die in absence of this gene. So we used this cabozantinib along with the inhibitor of our gene of interest or protein of interest, which is known as 1169E. And we combined them together and saw that yes, like the cancer cells were dying. and they were so how it works is you have to test whether like the drug by itself is effective or the drug in combination is effective. So we did both, and we saw as compared to drug by itself, the combination of drug is killing the cancer cells better as compared to like the single therapy. So that was really promising. Then we wanted to test whether you know what in terms of tumor models. So then I generated a tumor model, which is basically known as a spheroid. So what it is like generation of a tumor from a cancer cell line in the tissue culture. So it's like basically a spherical ball of tumor, which is now like it's gonna behave as a tumor. So then we did these tests again. So we had like drug by itself, we had the drugs in combination, and when we looked at the results, we saw yes, like the drug in combination is shrinking the tumor better than the drug alone itself. So that was that story, and I think like for the reason of like time constraints, we had to stop it there and then publish it. Of course, like the implication of this would be like next step would be like to test it in mice models or like the xenografts of human cell lines in mice. And I think like the really big thing that came out of this is like this concept of personalized medicine.
Medicine.
And how that works is like if let's suppose you have like a patient with cancer who has this gene overexpressed, now you can administer the other drug, the anti-cancer drug, but it will not respond because like the drugs are not working in absence of this gene. So the idea is like you would test a patient's like gene sequence first and see whether like you can administer the set of drugs on the patient or not, and what drug combination would be effective for that patient. So that's like, a beginning step for personal. Of course, there is a lot more work to be done, but like this is what like we think is happening.
Okay, so that majorly explains the transition from natural sciences to translational sciences as well. Seeing your discovery come to action, like would it take some 10 years maybe?
Well, I hope so because like people are still designing like novel inhibitors. And I think before my study, there was another study which tested like one of the inhibitors in prostate cancer, but it was the drug by itself. But this is like a study of its kind where so our chaperone inhibitor, the drug that I am using for our co-chaperone, it's by itself, it's not toxic to the cell. But when it comes in combination with the, accepted anti-cancer drug, it makes the cell sensitive. So I think that's the way going forward because like chaperones are very important and in the cell because they're highly they are required for housekeeping functions. So you cannot just go about and inhibit chaperones because your normal cells will die. So this strategy is basically using a co-chaperone inhibitor, which is a helper protein.
Hmm.
And what we are seeing is like if you affect this co-chaperone, cells are not affected as much as as they're sensitized. Sorry. So that's the strategy going forward. That rather than like just kill affecting the chaperone function, just inhibit the co-chaperone, which will sensitize the cell. You don't want like in terms of cancer treatment, you don't want the patient's normal cells to die as well because like that's going to have adverse effect on patient's health.
Yeah,
So I think choosing an inhibitor that like affects just the cancer cell is also like of importance.
sure. Hopefully, this is come to practice. And you did mention of CRISPR-Cas9, and this year
Mhm.
Dr. Jennifer and Dr. Emmanuel were awarded by the Nobel Committee for their work for their contribution to CRISPR. And women are starting to get recognized in their fields. So,
Mhm.
how does this make you feel as a fellow researcher in STEM?
I think like this was really exciting because we have been ever since I started doing CRISPR, we have been wondering when CRISPR is gonna get the Nobel Prize because like this really changed the way science works. So CRISPR has really revolutionized science. So I think like the one thing that I should mention is like before like the Nobel Prize, I think like the really exciting thing was like that came from like their company CRISPR Therapeutics was they treated a person with sickle cell anemia. So they using CRISPR. So they injected the modified, sickle cell anemic cell in hematopoietic system, and the patient was cured of sickle cell anemia. I think that was really exciting because now you can see like how CRISPR is changing lives, how you can use CRISPR to treat the patients. And I think that's the goal of every like, everything that we do in science, like how it impacts, like, how we can contribute to the society, how we can like help patients' lives. So that was really exciting. And then when we heard of like this Nobel Prize for Jennifer Doudna and Emmanuel Charpentier, I think it was really exciting. And of course, like we have been like anticipating like CRISPR getting a Nobel Prize, but like it finally happened, and it was really exciting. And I'm really happy that like they got recognized for their discovery.
Mm, yeah. So your insights would have added more value to this conversation. Thank you for joining us, Miss Nitika. Thank you.
Well, thank you so much for inviting me. I'm really excited. And
Yeah,
also, like, if you guys have any question about application to PhD program, just feel free to write to me. And I would try my best to answer your questions regarding, joining PhD or like how to go about it because I have been there. I know it's really hard because if you don't have anybody to tell you like how to go about the process, it can become a, really, but how should I put it? It becomes like, heavy on
even more
you.
tougher.
Yeah. Yeah, like you don't know where to start. And I have been there, so I know. So as I did not mention that, but like while doing masters, I wanted to go abroad, but I did not know how to go about it. So it can be hard. So if you have any questions, just feel free to write me. I will be happy to help.
Sure. Thank you. I'll stop recording.
Sure.